Hello and greetings from Plant Reactome team at Oregon State University. I am excited to share with some of the resources and tools we have for Maize community. So Plant Reactome is a plant pathway portal of Gramene. We use rice as a reference species to curate pathways and gene orthology to project on other species. Currently we have 97 other species including maize. We provide tools for data upload, visualization and analysis in pathway context. We also provide data services and APIs to access our data. Tutorials and training material related to Plant Reactome can be found here in the Outreach and Training portal of Gramene. You can access Plant Reactome by clicking here, or you can access plantreactome.gramene.org directly. Particularly for the Maize community, we have 253 pathways containing 637 reactions and close to 1900 proteins. You can click here on pathway browser and access all the pathways and listing that we have in the database. We also have right here the analysis tools available by analyze your data and the search option right here. I will show these one by one. So if you click here on pathway browser, you get by default into pathway browser window. By default, we show Oryza sativa, the reference species. On left hand side, we have pathway listings. On right hand side we have pathway diagram window, and here on the bottom panel we have associated information. Anytime you can access analysis tools. If you like to browse Oryza sativa, this is fine. You can also choose your favorite species such as Maize, and you get now the pathway listing for Maize that we have, and again the higher level fireworks view of the pathways. You can select a specific pathway such as salicylic acid signaling, which is very important in plant development and immunity and so many other processes. You get this pathway diagram of salicylic acid signaling. I would like to emphasize that our data model allows us to curate complexity of plant cell eukaryotic structure. We have many of such reactions depicted here occurring in the nucleus and remaining that you can see here in the cytosol. On the bottom panel then we have the associated information that I will show you in a minute. First I would like to show you a search option, so you can actually go back to Plant Reactome, and if you want to just search for a specific pathway, you can type your name, and here you can get again salicylic acid signaling or biosynthesis pathway. You click on the one that you like, and then you can directly get a link for the pathway, and you can come again in the same window. So there are two routes that you can get to a pathway. Here again you can explore these bottom panels. So description provides you the pathway summary and various citations that we have used for curation of this pathway. You can click on expression and you can select any genes from the pathway, and you can access the basal gene expression data that is available in the EBI gene expression atlas right shown here. Anytime you can access the analysis page. It allows you to compare your projected pathways with the curated pathway in rice, and here you can see that this projections overlay of maize pathways on rice pathways, and where you see these green boxes, it means that we have found maize and rice genes that are mapped on these reactions, and number of rice and maize genes are same. Where you find the boxes colored half blue or half green, then you find this combination or that combination. It means that number of the genes mapped to particular reactions in rice and maize differ in numbers. Again, these results are available for download right from here. You can go from one pathway to another pathway to visualize the projection of various pathways and their quality as well. Again, you can go to analysis space and you can upload any data that you have. It could be transcriptomic data, proteome, or metabolome data. So in this example, you see some of the genes upregulated, others downregulated, and you can actually click on a particular enzyme and you can see all the homologous genes and their expression profile. We have many duplicate genes in plants, and identifying the right candidate genes is essential for experimental analysis. So this tool is particularly useful for identifying the candidates and reducing the scope of the experimental research. Again, results and the mappings are available for download. So these are some of the tools that we have, and I hope that you find Plant Reactome as a useful resource for your research. We look forward to working with you, having your comments and your availability to review some of the pathways. I like to thank you and all the collaborators of the Gramene project at various institutes and NSF for funding. Thank you very much.